way possible, and really committed to working with marine science early on. And we have been fortunate enough to write recommendation letters for him to get into William & Mary, and he's excelled since then. And we hope to continue to observe his career as it rolls on. But it's a pleasure to present him to you, and I really hope that you appreciate this as much as we do, having seen him since 15, and you all seen him since he was. So
let me just say a quick word. For those of you who don't know, that's Russ Doctorberg, who did his doctoral degree with me, and of course Seth has been here for several years, and we are hoping he's probably going to have 2 or 3 publications coming from his high school and undergraduate projects. So quite productive, and of course we hope that he pursues his graduate degree at William & Mary instead of some university like Stanford or.
Thank you for that lovely introduction. Today I'm going to be defending my biology honors thesis, which is on the development of the habitat suitability index for the eastern oyster in the Great White River of Virginia. So to begin, I'll give a little background on the eastern oyster, which is the subject species of my research. So the eastern oyster is a bivalve mollusk, and so as adults, oysters live in benthic regions, estuaries, and they're sessile. So as adults, they don't really go anywhere. Where they've chosen to live is where they're going to live their life. And so as adults, they broadcast spawn their gametes into the water column, and they're externally fertilized in the water column, where they form into a free-swimming planktonic larvae that develops for about 2 to 4 weeks in the surface waters of the water column. And it's really at that point that the larvae are at the mercy of the currents. And so depending on the water flow in a particular estuary, they can either end up close to the reef of origin or very far from it. And so after that larval period, they begin to sort of settle towards the benthos, and they use chemical cues from other oysters to determine where they're going to settle. And so those chemical cues really hone larval oysters in on where to settle. And as they begin to settle, they tend to form into these sorts of clusters or gregarious reef-like structures where you have oysters settling on top of other oysters. And from an evolutionary standpoint, the reason for this is that by living close by to each other, since they broadcast spawn, they're able to more efficiently reproduce. And so there's other implications of this, through this formation of this three-dimensional gregarious cluster structure that have implications on other species and help to give them the status as an ecosystem engineer. But another sort of ecosystem service that they provide is their filter feeding. This may be the ecosystem service that you guys are most familiar with. This is because they take in a volume of water from the water column and they filter out both phytoplankton, suspended sediment, and other particulate matter from the water column. And by doing this, they reduce the sediment concentration in the waters, but they also are reducing the phytoplankton, the nutrients that are in the water. And so as you can see in this image here, we see an oyster taking in a volume of water. And so this is not trivial. They are able, an adult oyster is actually able to filter about 50 gallons of water per day. So this is a pretty amazing feat. And so this diagram really goes over some of these ecosystem benefits in a little more detail. So as I was saying before, the improved water quality is one of these ecosystem services that we're probably most familiar with. And this is really kind of twofold in that they are taking in phytoplankton, which reduces nutrients that are available in the water column. This is actually a pretty important thing in Chesapeake Bay, especially because eutrophication, excess nutrient loading into the bay is a real problem. And they're also removing sediment and depositing that in the form of pseudofeces, which reduces the amount of available sediment in the water column, reducing the concentrations of those particulates. But another sort of benefit of oysters and oyster reefs is that they provide substrate for hard-boiled benthos and refuge for other biota. And this includes species such as mud crabs, blue crabs, which are commercially important species, along with some other finfish and other species. So really in the absence of any other structure, such as obstructed bottom, which exists in a lot of Chesapeake Bay, these oyster reefs are providing a lot of these services. And so it's really important that there really is no other species in Chesapeake Bay that provides these ecosystem services at the same level as the native oyster in the bay. And so as you guys probably know a little bit about this, oysters in Chesapeake Bay aren't doing so well right now. But it wasn't always that way. And actually, prior to European colonization, oysters were so abundant in Chesapeake Bay that they actually posed a navigational hazard to ships, which is kind of crazy to think about. This quote from Captain John Smith really sums that up in saying that the abundance of oysters is incredible. So there are whole banks of them so that ships must avoid them. So this really gives you sort of an appreciation for just the abundance of oysters that once were in the bay. And so when European colonists began to colonize the Chesapeake Bay region, they relied on these oysters as a vital food source. And so as time wore on, as the colonies began to develop more efficient methods of harvesting these oysters as a fishery resource, as a food resource, we also saw the decimation of the bay's oyster population, mainly through the use of an oyster dredge, which is essentially a rake-like structure with a bag on the back of it that removes the top layer of an oyster reef. And as I said before, larval oysters rely on other adult oysters for substrate in order to have persistence of the reef over time. So in removing the oysters, you're not only removing the adults that provide the gametes to ensure the persistence of the reef, but you're also removing the habitat that's necessary for the reef to persist. And so as you can see in this graph, which tracks oyster harvest over time, there really was a pretty steady decline of the oyster population. And then in the 1950s, we had two major diseases, MSX and Dermo, that really served to further decimate the oyster populations in the bay. And currently in Chesapeake Bay, the oyster population stands at less than 1% of its historic abundance. And worldwide, oyster populations have been reduced to about 10% of their historic abundance. So this is not just the Chesapeake Bay serious problem, but really worldwide. And so in the early 1900s, native oyster restoration efforts really got kickstarted as people began to realize, we're losing this very valuable resource, very valuable fishery resource that supports a lot of the Chesapeake Bay region's economy. And so early efforts really focused on fishery enhancement, fishery restoration, and these involved shell plantings of oyster shell material, and this sort of a modern day example, but this is essentially how it's done, is you take out a barge filled with oyster shell and kind of chuck it off the side, and it settles out, and that's sort of how we restore reefs by shell plantings. And so this chart up here kind of shows, though, that historically fishery enhancement has been very economically inefficient. And it's also been very short-sighted, not really looking at the long-term enhancement of the oyster population, mostly looking at short-term fishery benefits. You can see this here between a 5-year period in Virginia with the shell subsidy program. There's a net loss of $8.5 million, so it's a non-trivial amount of money. But another thing to consider is the fact that there's a number of ecosystem stressors, such as nutrient and sediment loading, that persist, and they've been increasing over time with increasing development in the Chesapeake Bay watershed. So all these are things that are sort of hindering native oyster restoration efforts. Another key thing to mention is that historically oyster restoration efforts have focused on construction of what's known as low relief reefs, which are essentially just a thin veneer of oyster shell top to bottom. And in doing that, and having just a thin layer of oyster shell, that oyster reef is essentially very easily able to be covered with sediment and to basically not be able to persist in a very long-term fashion. So to contrast that, in 2004, in the Great Wycomico River, the U.S. Army Corps of Engineers constructed one of the largest sanctuary oyster reef networks in the world. And so the Great Wycomico River is about 20 miles south of the Potomac River, so some of you may be familiar with that area. But so in the Great Wycomico River, the Army Corps of Engineers constructed sanctuary oyster reefs. And so these oyster reefs were constructed not at all for fishery purposes, but in order to restore oysters for their ecosystem services, for the ecological benefits associated with them. And so in the Great Wycomico River, they used some fairly novel techniques, and they've achieved an unprecedented level of success with their restoration effort. And so one of the major things that they did was to construct the reefs using high vertical relief. So they used a larger volume of oyster shell, And so what they found with that is that those reefs that are constructed on using high relief reef techniques are generally able to outpace sedimentation, and that they're able to recruit a larger number of oysters and generally host a higher abundance of oysters due to factors such as increased flow rate, among other things. And this graph really tracks, and you can see just the differences between oyster density on the higher relief reefs versus low relief, which again is what was historically used in oyster restoration efforts versus unrestored bottom in terms of oyster density. So these higher relief reefs are very effective. And it's also important to mention that these reefs were constructed at a larger scale than most other restoration efforts. They encompass about 85 acres of river bottom, which is really an unprecedented scale for these restored reefs. And so these images show, this one on the far left shows Dave Schulte, who is a researcher with the Army Corps of Engineers and Defense. This shows one oyster from the reef. You can kind of see this gregarious cluster forming, and you can kind of imagine how with time that single oyster could really become a reef if oysters continue to settle and grow outward from that single oyster. And this other image just shows the large amount of live developed oysters living on the Great White Congo reefs. And this map is just intended to sort of show the distribution of the sanctuary oyster reefs in the Great White Congo River, just to give you an appreciation for the extent of these reefs in that river system. Okay. So in 2009, President Barack Obama implemented Executive Order 13508, which really focuses on the restoration and preservation of the Chesapeake Bay ecosystem. And one of the key goals of that executive order is to restore native oyster populations in 20 tributaries of Chesapeake Bay by 2025. And so in order to be successful in achieving this goal, we really need to be planning our restoration efforts very well as we can, using all available strategies to identify where the best areas are to be placing these restored reefs. We need to be using effective reef construction methods. We know that high relief reefs work, that they're very effective at recruiting a high abundance of oysters and sustaining that high abundance of oysters. And we need to be looking long-term at the monitoring of these restored reefs. And if a particular area in a reef system is not doing so well, we need to be going into those areas and rehabilitating them to make sure that in the long term, these restored reefs are successful. And we also need clearly defined success criteria. We need to be sure that we have a uniform description of what a successfully restored reef is. So to go into some of the detail on some tools that are currently used in oyster restoration planning, one of the most basic tools, I suppose, would be to use historic maps of oyster reef locations. And this image on the right shows the location of the historic oyster reef from a survey done in 1890 in the Glenhaven River system. So this can be useful to tell where oyster reefs used to be and where, maybe in the present time, where they could also be successfully restored currently. In this image, or this graph on the left, shows a connectivity matrix, which is the output of a hydrodynamic model, which allows us to see how restored reefs are connected. And it's important to know how reefs are connected by larval dispersal in order to ensure that reefs persist over the long term. Okay, but one key thing that's missing from the use of those tools is that we don't have an adequate idea of where suitable habitat, especially in this case, where suitable oyster habitat is currently. And so one method that conservation biologists have utilized to sort of solve this issue is through the development of habitat suitability indices. And so the main goal of these habitat suitability indices is to develop an index that quantifies habitat suitability in order to determine the areas of restoration and conservation interest. And just to give you sort of some perspective on the diversity of species these indices have been produced for, they've been produced for endangered species such as bald eagles, bobcats, and terrestrial ecosystems, as well as for Atlantic sturgeon and marine ecosystems. So some HSIs have been developed for oysters throughout the world. The first one was developed for the eastern oyster in Galveston Bay in 1983. They've also been developed for the Pacific oyster along the coast of Vancouver Island and British Columbia in Canada in 1988. And additional HSIs have been developed for the eastern oyster in the Hudson River, New York, the Maryland main stem of the Chesapeake Bay, the Caloosahatchee Estuary, Florida, and the Mission Aransas Estuary in Texas. But two main limitations of these HSIs that have been done is that they haven't been validated using independent data set. And this is important because it's great to produce some sort of model that tells you, where's best to be restoring, where's best to be conserving. But if you don't really know how valid that model is using an independent data set, it's not really all that valuable. And so this slide is also intended to sort of give you an idea of just the geographic spread of these HSIs and to give you the length of time. They've been in existence since about the 80s. So the goal of this research project, this honors thesis, was to develop an HSI for the eastern oyster in the Great Waikamako River. So the goal was to identify suitable oyster habitat in the Great Waikamako River in order to identify areas in the existing sanctuary reef network that can be rehabilitated, and to also identify locations in the river to construct additional oyster reefs. And so just to give a little more information about it, the goal also was to utilize recent high-resolution, so fine spatial scale data, GIS data in the model development, and then to validate that HSI with an independent recent oyster abundance data set to ensure that what we're seeing based on the use of GIS data is actually realistic, and that will give us a good idea of where actually the suitable oyster habitat is. And then also the goal is to really develop, the more general goal is to develop a model framework that can be generalized to any location in Chesapeake Bay, and also locations throughout the range of the eastern oyster, and also kind of looking more broadly, looking at developing a model framework that could be useful for maybe other oyster species in other parts of the world. And this slide just really goes over some of the steps involved in the development of habitat suitability index, and so we'll take them step by step. And so step one is really to define the variables that control habitat suitability. And so this involves a literature search to determine what those variables are, and then to develop species preferences. So this really involves a quantification of different variables on a scale of 0 to 1, 0 indicating a very low suitability for a particular parameter or variable, and 1 indicating very high suitability. And then the next step is once you have what those variables are, you need to determine how to actually get the data for those variables. So that involves data collection and then development of environmental layers within GIS or geographic information system. So for this habitat suitability index, it utilized 3 variables. The first of which was sea bottom type. And this was used or included as reefs constructed on rock or hard sea bottom types are far less likely to subside and degrade than those built on mud sea bottoms. And if any of you have ever been out in Chesapeake Bay on a mud bottom, you can tell just how that sort of bottom would be able to support a heavy weight or something. And especially considering how expensive oyster reefs are to construct, you don't really want to be building an oyster reef on something that's going to sink into and basically be lost. And so this graph on the left shows the reclassification scheme that was used within GIS. As you can see on the axes, we've got mud, clay, silt being given a value of 0, indicating very low, or actually unsuitable, all the way up to hard bottom or oyster rock, which is given a value of 1, which indicates very high suitability. And so this just shows you how I actually did this in GIS. So I took maps that had the distribution of various bottom types in the Great Waikamako River. So we've got purple, we've got clay silt, which is like a mud bottom, and oyster shell, which is kind of a pink color. And then I reclassified that according to these various bottom types in this reclassification scheme. And what we're left with is a map like this, which shows us where the highly suitable oyster bottom is, or highly suitable oyster bottom, versus the very unsuitable oyster bottom. And so high suitability is green, low suitability is red, with the yellow indicating the margin of suitability at the bottom. And then, so another variable that was included was depth, and this was included as reefs constructed at depths less than 12 feet. Host high densities of live oysters, depths 12 to 15 feet, host marginal densities of oysters, and then depths greater than 15 feet, host low densities of live oysters. And this was also verified by a sampling that I took over last summer in the Great Waikamako River. I went out with a DO meter and just measured the dissolved oxygen level at various depths throughout the river. That was also verified. This trend was verified using that. And this is due to seasonal hypoxia and anoxia events, which are associated with the decomposition of phytoplankton and its subsequent stratification of the water column, which I kind of mentioned earlier. So these figures are just showing the suitability reclassification scheme I used, and then the output variable layer that was used in the model development. And so the final variable that was included was salinity, and this was included as oysters can't tolerate very low or high salinities for long periods of time. And again, same sort of deal, we get the salinity reclassification scheme and the actual variable layer that was integrated into the model. And so we've got all those variable layers now, which is great. So the next step is really to construct a suitability model to sort of generate a generalized model that shows us based on all these variables that determine habitat suitability. It helps us to determine, where the suitable oyster habitat is. And so to do this, what I essentially did was to take a geometric mean of each of these individual variable layers. And the reason I did that was because if for a particular location, if any individual layer had a value of 0 for that location, you would not want that location to be considered suitable, because for that one variable it's unsuitable. So it's kind of confusing, but let me try to break it down. So if for one location you had, say, a mud bottom, but it was a suitable depth and a suitable salinity, you wouldn't want that area to be considered suitable. So that's why the geometric mean was taken. And so what you get after computing that in GIS is you get this map which shows us where suitable oyster bottom is. So again, greener areas of high habitat suitability, again based on all those variables. Redder areas of low habitat suitability, and then you've got sort of marginal suitability there in between. And so in order to validate this model, which is an important step, because again, it's great to have a model, but if it's not actually telling you what's really going on out there, it's not really useful. So in Chesapeake Bay, a live adult oyster density of 50 oysters per square meter is known as the minimum success criterion for restored reef to be considered successful. And so live adult oyster density derived from the 2011 DIMS monitoring survey of the Great Wacomico River sanctuary oyster reefs was used to validate this model. It's important to note that this data set was not at all used in the model development. It was fully independent of the habitat suitability index creation and construction. And so these are the graphs plotting habitat suitability versus the adult oyster density at particular sites. And so this is divided between high relief reefs and low relief reefs. And again, so high relief reefs are reefs that are constructed with a larger amount of oyster shell to make them essentially taller, whereas low relief reefs are just a thin layer of oyster shell and are fairly representative of historical oyster restoration efforts. And so what we see are some interesting trends in that for low relief reef, for example, we have areas where there's high habitat suitability. So 0.3 and 1. So again, one is high habitat suitability. We've got a number of oyster reefs that are in this range where they have high habitat suitability but low oyster density. So that really tells us that these locations could be suitable for rehabilitation of the Great Wacomico River. These could be areas where if we placed additional oyster shell material, we've got the high habitat suitability. In the long run, if we place that shell material there, we could see success in that location in terms of restoration. With the high relief reefs, we see at HSI about 0.5, we see adult oyster densities that far exceed the GIT target, the 50 oysters per square meter target. And so what this is really telling us is that if we build high relief reefs in areas that have an HSI value greater than 0.5, we're likely to see a lot of success there. But this also validates the importance of building reefs at high relief. Because it really shows that with high habitat suitability and high relief reefs, we're likely to achieve success in restoration. And so this map really is just breaking down and showing the habitat suitability index overlaying with the various sampling points that were used to validate the model. You can just kind of get an appreciation for how well the habitat suitability index and this abundance data set really line up. And so in order to maximize the success of oyster reef restoration and rehabilitation, to determine where suitable oyster habitat is, we've defined suitable oyster habitat as being all areas with an HSI value greater than 0.6, and marginal oyster habitat as being areas with an HSI value between 0.4 and 0.6. And so this map really shows the distribution of suitable oyster habitat in the Great White River, with green being areas greater than 0.6 HSI. That's essentially where the suitable oyster habitat is. And then the yellow areas being areas of marginal habitat suitability, again being between 0.4 and 0.6. So this is useful because the majority of the Army Corps are really in this area. And this gives us an idea of where, in more upriver locations, suitable oyster habitat is that we could build additional oyster reefs. And so this can be used with hydrodynamic model information and with more historic reef location maps to really determine and pinpoint where these best areas would be to restore in the Great White River. And it's also useful to identify where in the existing sanctuary oyster reef system areas could be that could be rehabilitated. And so that's part of long-term adaptive management to ensure that these existing reefs succeed well. And so this is just to show how this HSI information can be used in planning. It's also important to note that the current scheme I'm using to determine where suitable and marginal oyster habitat is really conservative. It could be that we can adjust the parameters to be more specific, and we will be working on that in the coming months. But so as you can see here, we've got in a low relief reef area, we've got a low oyster density and highly suitable habitat. So that would be an area where we could go in and place additional oyster shell material, and in the long term, it may end up being a very successful location in terms of being a restored reef. So there are limitations of the current HSI. This isn't all-encompassing. It's always going to be a work in progress. And so additional physical environmental variables may also control oyster habitat suitability. These could include water temperature. Oysters can't handle freezing water temperatures all the way up to very, very high water temperatures. Chlorophyll concentration, which is a surrogate for phytoplankton concentration. Very low phytoplankton concentration is bad because phytoplankton is their food, but very high concentrations is also bad because that could indicate a phytoplankton bloom that would ultimately lead to a low dissolved oxygen event through the decomposition of that biomass. So they're about marginal concentration again as well. Low pH is a consideration as acidic conditions reduce carbonate availability, and they also lead to the dissolution of shell material. Low dissolved oxygen is also factors as low DO can prove lethal. The frequency of cold snaps in the Great White River, there are intertidal oysters which are growing on hardened shorelines, and so this is a consideration if you would like to include that part of your conservation plan. As these intertidal oysters could be susceptible to very, very quick changes in temperature, very quick freezing events. And it may also be that there's a variable depth trend along the length of the river. And so what this means is that from the most downriver site near the mouth all the way to the river, there could be a different trend in the DO versus depth relationship. Whereas near the mouth, there could be enhanced tidal flushing, which would mean that deeper waters remain more well oxygenated than more upriver locations at the same depth. There's also anthropogenic influence variables that could control oyster habitat suitability. And these could include a probability of a juvenile oyster mortality event due to a freshwater influx. And that's really a function of adjacent land use. We could also, in the Great White River, sanctuary reefs appear to have been poached, which means that water may have either accidentally or intentionally drag their oyster dredges through these sanctuary reefs, which is really a problem because, as I said before, through removing the oysters, you're not only removing the live adults that are producing the gametes that ensure the long-term success of the reef in terms of recruitment of additional oysters, but you're also moving that habitat, which is really sort of a twofold issue. And another variable could be the likelihood of sanctuary reef success. And so those are all anthropogenic influence variables that could also influence habitat suitability. So during the spring of next year, after I've graduated, I'll be working to further develop this HSI, and one of the variables I'll be including is a variable depth trend along the length of the river. And so for this version of the HSI that I presented to you, I've used a uniform depth trend, D over C depth trend, but it is actually likely that waters greater than 15 feet depth near rivers now may be well oxygenated due to enhanced tidal flushing. And so when I actually went out over the summer, again, I noticed during neap tide where stratification is most severe, there was a trend, actually it's springtime. I didn't book it during spring, but so during springtime, we noticed that the waters deeper than 15 feet near the mouth were able to remain well oxygenated. So it'll be important to sort of pinpoint that relationship and to possibly divide the river into zones where we have different geophysical relationships. And so another variable will be a probability of the juvenile oyster mortality due to a freshwater influx event. And so that's a function of adjacent land use, and that's due to development lands adjacent to bodies of water accelerating the influx of freshwater. This map is just showing adjacent land use information, lakes adjacent to the river. It's derived from satellite imagery. And so we can see these red areas are areas of heavy development, and these yellow areas are areas of agriculture. So those are land uses that would possibly accelerate the freshwater influx and could lead to a juvenile mortality event. So it'll be interesting to include a layer like that, and of course they're useful. Additional development will include the development of population capacity derived from a hydrodynamic model. And I'll also use Martin conservation planning software to integrate ecological and socioeconomic information in order to optimize the distribution of sanctuary reef reserves that meets minimum ecological targets while minimizing relative social and economic ecological costs. So it's kind of a mouthful, but really what this tool is useful for is for allowing us to integrate social information, which is always important when you're looking at a conservation issue. Because conservation doesn't exist in a vacuum, there's always going to be social implications. So it's very important to look at that and to integrate information regarding those concerns and models. So sort of as an aside, another interesting thing I found during our research over the summer was a potential bottom use conflict between seagrass, which is a vital habitat in Chesapeake Bay of restoration interest, and aquaculture grounds. So through the Virginia Marine Resources Commission, public sea bottom can be allotted to private individuals in the form of a private shellfish aquaculture lease. And these private shellfish aquaculture leases are basically areas where the leaseholder has domain over their particular lease, and they can use it for oysters or clams or really whatever type of shellfish that they can grow for market purposes. And so within the Great Wycomico River, 36% of the available sea bottom is held in these private aquaculture leases. And so again, seagrass is a vital benthic habitat in Chesapeake Bay that provides nutrient buffering, food to waterfowl, and habitat for blue crabs, which are a species of a lot of conservation interests that you guys are probably familiar with. And so of the 311 acres in the Great Wycomico River, about 147 acres of that seagrass habitat is held within these private shellfish aquaculture leases. So this could be a conflict, and it's something that should be looked into further. It's just kind of something I came across when I was doing my research. And this figure just shows you the distribution of seagrass, which is in these sort of yellowish polygons, in relation to private shellfish aquaculture leases. And you can see a lot of that is really held within those leases. So something to think about. And this image from the NOAA's SAU lab shows the relationship between this SAV bed and its proximity to a shellfish aquaculture setup. So these can be very close to each other, and it's definitely something to think about. So regarding the future of Chesapeake Bay oyster restoration efforts, I think that the future is bright. I don't think it's as dismal as a lot of people probably think or maybe even want it to be. But so in 2009, the executive order by Obama established the goal of restoring native oysters in 20 tributaries of Chesapeake Bay by 2025. And so, if we use the same sorts of techniques that we're using in the Great Wycomico River, such as using really solid planning strategies, such as hydrodynamic models, massive historic reef locations, and then also this tool that I've developed, the Habitat Suitability Index, we can get a really good idea of where we can be. We can really pinpoint where we should be building these historic reefs, and then so now we know where we're going to be building these reefs. We can use the most effective reef construction methods, such as high relief. Reefs have been tried and true. They're a good method of constructing these oyster reefs. And then long-term monitoring of these reefs to ensure that they're successful and having clearly defined success criteria. If we do all of those things, I think we'll achieve a lot of success in restoring oysters in the bay. And so, sort of big picture, this Habitat Suitability Index framework can be applied to other tributaries of Chesapeake Bay. And it can also be sort of generalized to the distribution of the eastern oyster across its range, its geographic range, and then also to other oyster species. And another thing we need to think about is garnering public support for these oyster restoration efforts. These oyster restoration, these oyster reefs provide a lot of ecosystem services, a lot of ecological benefits to the Chesapeake Bay, and a lot of people are interested in saving the bay. And this could be a real way of doing that, and so we need to be able to communicate this to the public that communicate these successes in restoration. And so that's pretty much it. I've got some acknowledgements, a lot of them actually. First, I'd like to thank my parents, family, and friends without whose support I wouldn't have been able to do this thesis and definitely not been able to finish my college education. I'd like to thank Dr. Matthias Lloyd, Dr. Ron Lipsius, along with my thesis committee for spending so much time working on my thesis with me and providing a lot of help and support along the way. The VIMS Marine Conservation Biology and Community Ecology Lab for giving me a lot of chances to get away from the computer and to do some learning, which is much needed during the summer. And then Dr. Jim Shedd of VIMS and the NOAA Chesapeake Bay Office of Habitat Assessment for providing GIS data. And then also the Army Corps of Engineers Norfolk District, the William Mary Center for Geospatial Analysis, and the William Mary Roland R. Charles Center for providing funding and for just a lot of assistance along the way. So that's it.
Other questions for Seth? Okay.
Yeah.
So I had two questions. Pretty much kind of get it. The first is how much total area did your HSI predict as good or at least end marginal habitat?
I've got those numbers in my thesis, and I was considering including them, but I didn't want to bombard everyone with numbers. I'm trying to think offhand. I'm thinking it's in the hundreds. So I'd have to
Okay, here's the
exact number, but I could tell you.
171 acres.
Okay, there we go. I should probably know that.
And is there a minimal area that's necessary for long-term reef persistence? Do you know?
I don't. I know a lot of previous efforts have been really patchy and just building small reefs and hoping that that small reef would take off, I guess. And so in that regard, I don't think that's a very successful restoration strategy. But really what you'd want to do is to create a system, a network where they're connected by a larval dispersal. I don't know that size is necessarily important, but I know that you wouldn't want it to be too small, but you want it to be large enough to be able to persist to outpace sedimentation. So in terms of an actual minimum size, I know that that's sort of a debate in ecology, is this the single largest settle small debate. I don't know an answer to that, but I know that by building several small, you kind of are able to maybe wither stochastic events. With one part of the network it's taken out by some sort of disturbance event, the other parts will still be okay. So there's not a management. Hopefully that answers a little bit.